Sounds good. Yeah. Thanks. Yeah, thanks so much for the kind introduction. It's always so, yeah, as you know, I did my PhD here at Berkeley, and it's always wonderful to come back here and see the vibrant community. You know, many of my mentors are sitting in the audience. You know, Petter was my PhD advisor, and the first neuroscience class I ever took was taught by Bruno and Frederick and Fritz. So it's really fun to be back here, and it's wonderful to see the community come back together after the pandemic. So yeah, so you know, it's kind of funny. I was doing some post hoc analysis of the title that I chose for this talk with Na, and you know, trying to have the title appeal to a physics audience. So just if you're reading the title, just look at the words neurophysics, emergence, collective dynamics, and interacting. Right? Those are all physics words. Don't let the little cognition and neurons off-put you too much. All right? We'll try to sweep them under the rug. But in any case, so here are three brains, right? They're immensely complicated, really large, vast networks of interacting neurons, right? And so how can we hope to possibly understand these systems? Well, on the experimental side, experimental neuroscience has made remarkable progress in terms of our ability to probe and perturb these systems. For example, we can now routinely record thousands of neurons simultaneously in, say, the brains of mice. And moreover, we can even write into the brain patterns of neural activity through optogenetic stimulation on the order of magnitude of hundreds of neurons. But this is still orders of magnitude less than the size of the relevant brains. So even experimental neuroscience, despite its remarkable progress, has a significant undersampling problem, right? On the other hand, advances in machine learning now allow us to generate highly complex, veridical, accurate models of neural circuits, accurate in terms of being able to predict variance in data. But this raises a profound question. Are we simply replacing something we don't understand, i.e. some part of the brain, with something else we don't understand, a highly complicated model? So statistical and computational neuroscience has a major understanding problem also. Right? And so we want to try to crack this undersampling and understanding problem as much as we possibly can. And so some of the work that we've been doing can be characterized as sort of two related thrusts. One is developing principled theories to guide the design and interpretation of large-scale neuroscience experiments in the undersampled regime. And the other is to advance explainable artificial intelligence to both understand complex models and extract testable hypotheses from these complex models. And of course, these are intimately related questions. Confronting what it means to understand how distributed nonlinear circuits compute can help us determine what we should indeed measure and manipulate in order to most efficiently arrive at that understanding. And so here are some sample works in kind of both thrusts. Of course, artificial intelligence also has an understanding problem, right? We have these amazingly complicated neural networks. We'd like to deploy them in society, especially in situations where we have regulatory risk and so forth. We really need to understand how they work and what they're doing. And actually, it turns out physics can help quite a lot here. So in another thrust of research, we've been looking at just artificial neural networks and trying to understand how they learn, how they work, and so forth. And it turns out there's a lot of room for ideas from physics, including non-equilibrium stat mech, dynamic mean field theory, statistical mechanics of random landscapes, and other aspects of mathematics to shed conceptual insights into AI. It turned out I actually gave the Berkeley Physics Colloquium way back in 2000, I think in 2015. And then I talked about our work on deep learning theory. So just in order to not bore anybody, I decided to focus more on neuroscience in this talk. But if you're interested in this subject of deep learning theory, we recently wrote a review article in the annual reviews of statistical mechanics that outlines, hopefully in a somewhat coherent way, a wealth of work on the theory of deep learning from a physics perspective. So, if you're a physicist, this might be a fun place to start. Also, in terms of high dimensional data analysis and complex neural systems, we also wrote an older review article that shows how to use the replica method in a unified way to attack a whole bunch of problems in machine learning. And that might also be of interest to sort of physics grad students and so forth. Okay, but what will I actually talk about in this talk? I'll talk about sort of 4 vignettes, and I'm going to try to stay kind of at a high colloquium level and not go into deep technical details or anything, but I hope I'll be able to communicate stuff at a high enough level. We'll see if I succeed. So first I want to ask, what determines fundamental limits on our acuity to detect shapes, right? These could be really bright objects, for example. So the number of photons coming into our retina is not a bottleneck, but we sometimes have a hard time still telling the differences between shapes. Why is that? And what's the mechanistic origins of this limit? The second is, how sensitive is our cortex, the surface of our brain, to external stimulation, and why? Okay. And then the last two are a little bit more on the explainable AI side. Why does the retina have the diversity of cell types that it does? And why might medial entorhinal cortex contain these famous hexagonal grid cells? And why are they so prominent in so many different species? Okay, so hopefully this provides a nice mixture. I chose this selection of problems because ideas from physics play a role in solving them. All right, so fundamental limits on our ability to detect differences between shapes. So here are 2 oriented gratings. You can see that one is slightly to the left of vertical and one is slightly to the right of vertical, and you can sort of tell the difference, right? If you bring them a little bit closer together where the delta angle is, say, 2 degrees or 1 degree, it's going to be very hard for you to tell the difference. Okay. Why is that? This is a puzzle, actually, because if you show an oriented grating to an animal, say a mouse, tens of thousands of visual cortical neurons respond to these stimuli. Right? If these neurons have independent noise or make independent errors, then if you can average over their errors, your standard error in the estimate of angle should be 1 over the square root of the number of neurons, right? Classical measurement theory. So your just noticeable difference in angle should be order of magnitude 1 over the square root of the number of neurons, say 10,000. And this should be 0.005 degrees, right? But we or mice can come nowhere close to that. We're orders of magnitude worse. Why is that? Okay, and natural conjecture is the one assumption that I've made so far that these neurons are non-interacting or have independent noise might not be true. The conjecture would be that correlated errors dramatically limit stimulus information in the brain. Okay? But how do we show that experimentally? So my colleague in applied physics, Mark Schnitzer at Stanford, and the first author, Oleg Rumyantsev, they built this remarkable microscope that allows us to image thousands of neurons across the visual cortex of mice while they're just passively watching 2 stimuli. So we did the experiment where we showed the mouse 2 oriented gratings and we recorded thousands of neurons. Okay? And we'd like to understand the structure of signal and noise about the stimulus in this large population. So the bottleneck is not how many neurons you can record now, it's how many trials you can show to the mouse before it becomes disinterested. And you can show maybe hundreds of trials. Okay, so if you think about each trial as recording a vector or pattern of activity across 1,000 neurons, your data set is fundamentally, say, a few hundred points in a 1,000-dimensional space of neural firing rate patterns, right? The activity levels of each vector, of each neuron, right? Now, if we look at the correlational structure across trials, so this is the average pairwise correlation coefficient between one neuron and another neuron, these individual pairwise correlations are weak. Their average value is about 0.05, the correlation coefficient. But remember, we only have about order of magnitude 100 trials, so the error in the estimate of any individual correlation coefficient is 1 over the square root of the number of trials, so say 0.1. So how can we measure information in the brain, and especially the impact of correlated noise on this information, if we can't get any individual correlation coefficient correct? And moreover, there's n choose 2 possible correlation coefficients, and we're getting all of them wrong, where n is the number of neurons. Okay? So let's think about this a little bit geometrically in higher n-dimensional space. So imagine that this is an n-dimensional space where each axis is the firing rate of individual neurons, right? So this is the firing rate of recorded neuron number 1, firing rate of recorded neuron number 2, 3, all the way up to n. Okay? Imagine we show stimulus A once, we get one pattern of activity. We show stimulus A again, we get another pattern, and so forth. And then we show stimulus B, we might get one pattern here, stimulus B again, we get another pattern here. We never get the same pattern twice because of variability in the brain, right? And the information is roughly the signal to noise ratio, where we're actually working with something called Fisher information, but you can think of it very simply as the noise ball, right, for all the responses to stimulus B has a certain covariance ellipsoid, right? And it has a certain mean. The noise ball for the red stimuli, stimulus A, has a certain mean and it has a certain covariance ellipsoid. They're typically similar covariance ellipsoids. The signal is the distance squared between the means, and the noise is roughly the width of the covariance ellipsoid along projected onto the signal vector, the vector from one mean to another. So roughly you can think of Fisher information geometrically in this way, right? Signal as a length divided by noise as a length along a certain dimension, right? Now, what happens is, as you increase the number of neurons, the theory would be, if correlated noise is so strong as to fundamentally limit perceptual acuity, what should happen is, as you increase the number of neurons, you get more and more views of the two stimuli. So your signal increases proportionally to the number of neurons. But the noise must therefore also increase proportionally with the number of neurons. The ellipsoids spread apart as you record more neurons, but their covariance ellipsoids also grow, so that the signal to noise ratio saturates to something that's order 1. That would be the conjecture. Okay. But then this begs the question, we can't get any individual correlation coefficient correct. So can we even get the signal and noise, i.e. the particular covariance ellipsoid and its primary orientation along to the signal direction, can we get those correct in a regime where we can't get any individual pairwise correlation correct? And so I worked out a mathematical theory for how well we should be able to estimate this Fisher information in large neural populations. The ideas I used were random matrix theory, and there were certain phase transitions in random matrices that played a role. I won't go into those details, but I'll give you the take-home message because it suggests an answer that runs counter to conventional wisdom, right? Conventional wisdom might say, if I'm recording thousands of neurons, that I better measure proportionally thousands of trials in order to be able to accurately leverage my large-scale recordings, right? More neurons and I need more trials. It turns out the complete opposite is true. So here's the output of the mathematical theory that shows that, so what are you looking at here? This axis is the number of trials over which you take recordings. Each trial is showing one stimulus. This axis is the number of neurons that you record. And you're always going to underestimate the information, it turns out. And this ratio tells you how much you're underestimating it. So a 1 is good, that means no underestimate. So red is good and blue is bad. You're underestimating it severely. And you can see that isocontours of constant performance turn out to be hyperbolas in a plane formed by the number of trials and number of neurons, such that if you record more neurons, you actually need fewer trials to get the information correct, right? The intuition for that is the following. Each neuron gives you additional information about the axis of this covariance ellipsoid. And so it gives you another window into the structure of the covariance ellipsoid and the direction of the signal, such that if you add more and more neurons, you add more dimensions to the problem, so you don't actually need that many samples to get the covariance ellipsoid correct. That's intuition. Mathematically, what's happening is you're trying to estimate the principal eigenvector of a random matrix, where the principal eigenvalue or the signal is growing linearly with the number of neurons. Therefore, you don't need that many samples to estimate it because the signal that you're trying to estimate is growing with the number of neurons. That's a technical detail, don't worry about that. But the basic take-home message is that this theory reveals that the countervailing wisdom in neuroscience is actually the opposite of what you might expect. You can record many, many neurons, and then you don't need that many trials to get the right answer about collective emergent properties of the circuit, like the information in the circuit. So everything boils down to the true value of the signal to noise ratio, right? So the true value of the signal to noise ratio plays a role, and that appears as a parameter in the theory. We fit the parameters of our theory to the estimated signal to noise ratio in the brain. So these are actually curves that we would expect in the experiments that we did, right? But yeah, the particular stimuli only show up in the theory through the signal to noise ratio. Yeah. Yeah, so this works. Yeah, so Fisher information, if you have a continuous manifold of stimuli, then you have to worry about the gradient of the response with respect to small motions of the stimulus. So you could generalize it that way. But yeah, the Fisher information is fundamentally about discrimination between two stimuli, roughly, two nearby stimuli. Right? You could think of it more generally as the KL divergence between the conditional distributions of the response conditioned in stimulus A and stimulus B under a Gaussian approximation. That's sort of what we're working with analytically. Okay. Now, what's the actual answer in the brain? So we measured the information, and we found that it did indeed saturate. And it saturated already at about 1,000 neurons. So despite the fact that 10,000 neurons respond to the stimulus, if you pick any 1,000 neurons, you'll pick out the identity of the stimulus as well as can be expected. Recording more neurons than that doesn't help. We made sure that this was correct because if we subsample the number of neurons and number of trials, we get the same answer. And moreover, to show that noise correlations are what's limiting the information, we created a surrogate data set where we trial shuffled the data. We created these fictitious trials where the activity of neuron 1 comes from one trial, the activity of neuron 2 comes from another trial. So these are surrogate null data sets that preserve all single neuron statistics, but completely destroy pairwise statistics. And then how does the information scale with the number of neurons? It scales linearly, right? So as expected for independent neurons. So the signal to noise ratio is growing linearly, which means you'll have exponentially small error probabilities in the signal to noise ratio, which is what you would have expected, this sort of 0.005 result. But instead, we get something that limits perceptual acuity to about 1 degree in mice. Okay. All right. Now, it turns out that this feature that if you record more neurons, you need fewer trials, turns out to be quite generic in a whole bunch of settings. Basically, whenever multineuronal activity patterns exhibit some kind of low dimensional structure, we will see the same trade-off, the same beneficial trade-off between neurons and trials. This is fantastic news for the future of systems neuroscience because it suggests that, over time, we know that microscopes will only get better and better, that animal stamina will not get better and better over our lifetimes. So as we increase the number of neurons, we don't necessarily have to rely on animals that can concentrate for longer. In fact, we can rely on fewer trials and still be okay. This shows up in discovering precise spike time. These are all works that we've done in discovering state space dynamics and extracting learning and cognition and so forth. I'll talk a little bit about the state space dynamics just to advertise one result, just to drive home this point. Okay, in neuroscience, what you do is you stick electrodes into the brain that measure voltage deflections in the vicinity of the electrode, right? Now, near an electrode, there might be many, many neurons that have their voltage imprint on the electrode, right? So then you have this complicated sorting problem, right? Each electrode is listening to multiple neurons, so you have to sort the individual neurons and discover their identity, right? You usually solve this by some kind of clustering algorithm in the shape of waveforms that each neuron imprints on an electrode. This is an error-prone process. It's incredibly time-consuming. It's a major bottleneck in systems neuroscience. Okay? But oftentimes, what we do is once we get the single neuron activities after spike sorting on the electrode, we just do dimensionality reduction to visualize the state space dynamics of the circuit. And we often make scientific inferences based on these low dimensional state space dynamics, right? Could we skip the spike sorting step, right? Instead of taking the neurons, doing the spike sorting, doing the dimensionality reduction, looking at the neural trajectories, making scientific inferences about how that particular circuit works, could we just skip the spike sorting step and just get neural trajectories directly from the electrodes themselves? Right? In this paper, we showed that the answer is yes. And we relied on ideas from the geometry of random projections of manifolds, right? I don't have time to go into that, but if you're interested, we wrote a review article quite a while ago on this. The basic idea is the set of neural activity patterns that might occur in an experiment don't fill up the exponentially large volume of all possible neural activity patterns. You usually get a much more restricted set that might lie on some low dimensional curved manifold. And if you randomly project that activity, say from neurons to electrodes, or maybe from all the neurons to a subset of recorded neurons, the geometry of this manifold might be quite well preserved under this random projection. And we can develop theories for how this manifold will become distorted as you go from the original space to the projected space, whether it's from neurons to electrodes or all the neurons in the brain region to a subset of neurons. Okay. And so we applied this random projection theory to, for example, real simulated data and data from actual monkeys performing various tasks. And we asked, this is an example where these are trajectories from spike sorted data, and these are trajectories from unspike sorted data, and you can see they're qualitatively similar. We can compute the distortion in the all pairwise distances between this set of trajectories and this set of trajectories So a distortion of 0.1 is good, a distortion of 0.5 is bad. And we can ask, how does the distortion scale with the complexity of the trajectories? There's three particular aspects of the complexity of the trajectory that play a role. One is the duration of the trajectory, this parameter capital T. One is the autocorrelation time of the trajectory. Roughly how long do you have to wait for the trajectory to curve and explore a different dimension? That's tau. And the number of trajectories, that's C. What happens is we predict that the distortion will scale logarithmically with the duration divided by the autocorrelation time, and logarithmically with the number of trajectories, and inversely with the number of electrodes. The distortion should scale linearly, or you should see a scaling collapse of these curves here if you plot them as a function of just this combination of variables. And we see both in simulations and in actual data from monkeys, this predicted collapse. And you see that really all that matters for how distorted your inferred state space dynamics is depends on just these three parameters in this particular combination, or four parameters in this particular combination. So again, the same story that I talked about for estimation of signal and noise or Fisher information holds for the estimation of the geometry of trajectories. If things are roughly low dimensional, i.e. not that many trajectories, not that wiggly, not lasting for too long, then you don't need to record that many electrodes or neurons to get the state space dynamics correct. Yeah. All right. So basically, emergent low dimensional structure in the brain is quite easy to get, despite not being able to record all the neurons in any particular brain region, despite being in an incredibly undersampled regime experimentally at the moment. Okay, let's shift gears a little bit to now leveraging. See, one of the amazing things that's happened is we now no longer have to just passively record from the brain. We can actually stimulate the brain as well. Okay? So what can we learn from this optical read-write technology? Okay? So again, I'll explain it at a very high level. So this is a collaboration with Carl Dijkstra's lab led by Tim Marshall. So here what they did was, again, the behavior is very simple. They're asking a mouse to discriminate between two stimuli, right? For example, for a vertical stimuli, they ask a mouse to go or to lick a certain reward port. For a horizontal stimulus, they ask a mouse not to lick, and if it does lick by mistake, it'll get an air puff. So we're training up a sensory motor contingency between two images and two actions. The purpose of the two actions is to read out at a behavioral level the mouse's percept of the two stimuli. Okay, while the mouse is doing this, of course, I mean, the incredibly talented colleagues in Carl's lab created a microscope that could optically image calcium activity in the mouse's visual cortex, about 5,000 neurons simultaneously in visual cortex. Okay. We can learn the response properties of these neurons. How selective are they for a vertical grading, and how selective are they for a horizontal grading? So you get two populations of neurons, one that fire for a vertical grading, but not for horizontal, and another population that fires for horizontal, but not for vertical. So we sort of learn the language of the brain in some sense. Then we ask, do you really need all these neurons? Can we slowly take away the stimulus and replace it with a fictitious stimulus by writing directly into the brain? On some trials, we might stimulate some number of neurons that would have responded to the vertical grading. On other trials, we might stimulate some number of neurons that would have responded to the horizontal grading. Okay, so we basically replace the stimulus with direct patterned optogenic stimulation corresponding to a subset of so-called isotuned neurons that are tuned the same way to the stimulus, either vertically or horizontally selected. We observe the behavior and we record neural dynamics in response to this direct cortical stimulation. And these are the results. This is behavior. Notice the horizontal axis is the number of neurons that we stimulate, right? Going from 0 to about 40. You can see that there's a sharp change in behavior. This is the percentage of trials that are correct. Basically, at around 10 neurons, the mouse doesn't do very well, but at 20 neurons, it does exceedingly well. This is remarkable, right? You can stimulate 20 neurons in the mouse's brain, and it acts as if it's seeing the original stimulus, or at least its behavior is as accurate as if it were seeing the original stimulus. You can also record from the 5,000 surrounding neurons to the 20 or 40 or whatever that you're stimulating, and you can ask what percentage of them are responding. And you see that there's quite a sharp rise. Around 10, not many are responding, but around 20, you get a just noticeable difference. And around 40, about 30% of the network is active, right? 30% of the neurons that should respond are active. The neurons that shouldn't respond are actually not as active by any means, right? So basically, stimulating about 40 neurons stimulates about 30% of the relevant subnetwork in the brain. So the brain is incredibly sensitive. Stimulation of as few as 20 neurons, but not 10, is sufficient to trigger both correct behavior and widespread recruitment of a large fraction of isotuned neurons. Okay, now as a physicist, we'd like to understand why. Why is the brain this sensitive? Why is it not more sensitive? Can we understand what's going on? Mechanistically, how can this happen? How can you have this exquisite sensitivity to external stimuli, but still have low levels of stable spontaneous activity? Okay, so again, I'll just, at a very high level, so what is the functional significance of this observation? Well, the brain has low levels of spontaneous activity. If we just do a back of the envelope calculation, assuming this activity is Poisson and roughly independent across neurons, then you can calculate large deviations of this spontaneous activity. And you can ask this threshold of 20 neurons, how many standard deviations is it above the random noise in the system in the spontaneous state? And it turns out it's only about 4 standard deviations above the strength of fluctuations in spontaneous activity. So basically, this threshold for what we call ignition of the circuit and ignition of behavior is just high enough that spontaneous activity has a very low probability of generating false positive perceptual events. So basically, the brain is as sensitive as it can possibly be without having false positives generated. Okay, now what kind of neural network can achieve this exquisite sensitivity while retaining stable and low levels of spontaneous activity? So we developed a mathematical spiking neural network model of integrated fire neurons that could actually recapitulate all of this data. And the key idea is you have an excitatory subset of cells that all are tuned to the same stimulus and an inhibitory population. The excitable population by itself is unstable, but when it's coupled to the inhibitory population, it stabilizes it. And the inhibitory population is slow to respond, but the excitatory population is fast to respond. So then if you stimulate a small number of excitatory cells, they explode, they drive the inhibitory cells, and they give delayed feedback that shuts down the excitatory cells later. So it acts like a prototypical example of an excitable system in physics. The dominant model of cortical activity, namely this balanced excitation inhibition, can't actually account for this data. Now, let me go a little bit into more detail into the model because it actually makes a very interesting prediction about the structure of spontaneous activity before the stimulus comes on. So again, this is schematically the system. Here's a network of excitatory neurons, a network of inhibitory neurons. These excite the inhibitory neurons inhibit the excitatory neurons, and there's a external input coming in. So this network has a phase diagram, right? In a 2-parameter space, where 1 is the relative ratio of the strength of inhibition to the strength of excitation. The other is the strength of spontaneous inputs coming in. So that's this vertical axis, and this horizontal axis is the ratio of inhibition to excitation. And there are phase boundaries here, where with weak external input, the system is quiescent, but here it undergoes a saddle node bifurcation, and here it undergoes actually a Hopf bifurcation. This is what we figured out by analyzing the spiking neural network using Fokker-Planck theory. And these boundaries are actually obtained through a numerical analysis of our Fokker-Planck equations. So here's a critical line between a quiescent and an active or oscillatory state, right? And we could ask, so in order to explain this data, we need the network to be poised near this bifurcation so that a very tiny amount of input will push it over the edge and yield a very large response. So we can ask, what is the structure of spontaneous activity in neural networks that are poised near this bifurcation point? Okay. It turns out you see avalanches in the spontaneous activity, where by avalanche, I mean a set of neurons that are active in a really short time window, and then it stops. And then another set of neurons are active in a time window, and then it stops, right? So these avalanches have two relevant order parameters. One is the size of the avalanche, the number of neurons that are active from beginning to end of the avalanche. And T is the duration of the avalanche. And the distribution of sizes and durations both obey a power law with two different exponents. It turns out that these two exponents are not independent of each other. They actually obey a linear relation. And so there's a one parameter family of possible exponents, and which particular point in that one parameter family you get depends on where you are along this line. And so I'll show that to you. So here's the one exponent, here's the other exponent. This is a theoretically predicted line. And this is, so you get different types of avalanches if you move along this bifurcation. Right? We were actually very happy when we got this because, believe it or not, this is already experimentally observed. So this can be viewed as a post-diction of a phenomenon that had not been explained before. So here are actual data from avalanches measured in freely moving mice, in turtles, in anesthetized monkeys, in anesthetized rats, in slice, and so forth. And you can see that these two exponents are not independent of each other across species. They vary across species, but they vary in this strongly correlated manner. And this is exactly what comes about from our theory. And so the take-home message is a one-dimensional phase boundary in excitatory inhibitory spiking neural networks explains the existence of a one-parameter family of avalanches across multiple species. And that's a natural consequence of being a so-called dynamically critical network poised at this bifurcation, ready to be sensitive to small inputs. Yeah. They're leaky into green fire. Yeah, this G is actually a dimensionless parameter that takes into account the leakage and all that. But there, you can non-dimensionalize the equation so that roughly there's these two important parameters, and the leak, of course, shows up inside them. Yeah. Yeah. So, okay, good question. So, does this apply to any region of the brain? Okay, so let me tell you a little bit more about the theory. So what did I assume about this? So this is an excitatory subnetwork. So there's lots of neurons in here. And I assumed that the connectivity was Erdos-Renyi. So it's just a completely random connectivity. There's no structure in the connectivity. Same here. This is an Erdos-Renyi network of inhibitory neurons. And these neurons are random and these connections are random. So there's no other structure. So this applies to random networks that have no other structure above and beyond, right? So if other regions have other structures, then all bets are off, right? In our applications of the visual cortex, we think that anyway, so hopefully that answers your question. Okay. This avalanche distribution, or this relation between exponents, seems to be a universal property of excitatory inhibitory connections with Erdos-Renyi connectivity. Okay, so just take a message. You know, the cortex seems to be organized such that you have this sort of low levels of pitter-patter of spontaneous activity, but it's highly nonlinear, unlike the surface of water, in which if you stimulate just the right subset of cells and you activate them just above the pitter-patter of spontaneous noise, you'll generate sort of a tsunami of activity in cortex. And to step back a little about where I think the future of systems neuroscience might be going, is we're going to see more of this type of work where we take an active approach to both recording and probing the brain. So with this optical read-write technology, we can hopefully learn the language of cortex, i.e. how stimuli relate to behavior. We can design stimulation patterns that modify this language. We can play back this modified language to cortex, and we can learn something about how it works by both observing its response, both in neural activity and behavior. And so this is probably the simplest version of this much more general program that I think will happen. All right. I'm going to skip the retina, and we'll come back to it if there's time. I'll go directly to hexagons because pattern formation is part of physics. So let's talk about that. Okay, so let me skip the retina. We're short anyways. Okay, so computational origin for grid cells, right? So this is work done by these great students and a postdoc. It's in these two papers. So, and this is about to appear in Neurons soon. So grid cells are this famous sort of Nobel Prize winning discovery along with place cells. So these are cells in an area of the brain called the medial entorhinal cortex, deep in the brain, far from the sensory motor periphery. And this is what a grid cell looks like. So what you're looking at is a 2D enclosure that a mouse has explored. Okay? And this heat map represents the firing rate of a single neuron averaged over all times when the mouse visited a particular location in this 2D arena. Red means the neuron fired a lot, blue means it didn't fire very much. So as you can see, this one cell likes to fire if and only if the mouse is at the vertices of a sort of a hexagonal lattice. So, you know, this is a kind of a beautiful structure. They've observed the structure not only in mice, but in rats, in bats, and even in humans through fMRI while they're looking as a function of gaze position, not as a function of where you're moving around. Yeah. Okay. So the natural question is, who ordered these things, right? Why did evolution construct grid cells? What computational purpose might they serve? Yeah. Oh, excellent question. Yeah. Yeah, I wasn't going to talk about it, but that's an excellent question. So you get these grid cells in modules. So what happens is you might get one grid cell that's characterized by particular orientation and wavelength, the lattice, right? And then all the other cells will have the same orientation and wavelength, but with the translates of each other, right? And all of these cells sort of talk to, or we think they talk to each other. But at least the experimental data is you get these modules that are characterized by orientation and wavelength, and then they're translated. Okay, so yeah, they're translated with structure, and then you have another module with a different wavelength, but actually the orientations tend to lock to the boundaries of the box. Okay, so it's thought that one of the purposes of this medial entorhinal cortex is to path integrate, right? So how do I know where I am? One way to figure out where you are, or for the brain to figure out where you are, is to integrate velocity to construct an estimate of position. That's the problem of path integration. So let's test whether path integration alone could be that function, or would asking a neural network to path integrate lead to the automatic emergence of grid cells? Okay. Now, we weren't the first to try this. Other people tried to train neural networks to path integrate, and they got mixed results. This work got square grid cells in a square environment. And this work actually got quite heterogeneous grid cells in a square environment. So they didn't quite get hexagonal grid cells in a square environment. And they completely left open the question of why they even got square grid cells to begin with. There was no theoretical explanation. So the questions we asked were, when and why do grid-like representations spontaneously emerge in neural networks trained to path integrate? If grid-like representations emerge, what determines their lattice structure? Square, heterogeneous, or hexagonal? Can we find trained path integrators that reliably yield hexagonal grid patterns? And if so, what generic circuit mechanisms give rise to these hexagonal grid patterns? In essence, can we sort of do this explainable AI? Can we go from a complete connectome and brain activity map to a conceptual understanding of network mechanism? And what procedure would that look like? Okay, so what we did was motivated by some other work from Dori Dederikman, we decided to put a little bit of biological realism into these networks, and a very simple thing. So we trained a neural network. We actually simplified the neural network compared to previous work by DeepMind. And so we had a recurrent neural network here with a trainable connectivity J. We had velocity input as a 2D vector through trainable weights M. And we had trainable readout weights W. And that created a so-called place cell code, i.e. a set of neurons where we want them to fire if and only if the mouse is at a particular location. And you have a population of these for every location. And the basic idea is we just feed the network the velocity, and we feed the network the correct positional output, and we train all the connections here to solve that problem. Okay, we're not claiming this is a model of how the system develops because we're providing teacher inputs to the system, but this is just a method to explore in an unbiased manner the space of neural networks that path integrate. We provide that as an input to the system. So there's an initial condition that basically resets the network to the appropriate location. Okay, so okay, so what happens to the firing rates of the hidden units as the network, as the agent explores the environment after it's been trained? So if we force the hidden units to only have positive firing rates, for example, by having rectifying units, then it turns out we get these hexagonal grid cells, along with some heterogeneous cells. If we don't force the neurons to have positive firing rates, it can have both positive and negative firing rates, we get these heterogeneous cells that look a lot like what DeepMind got. But if you do a proper statistical test and you compare this to low-pass filtered noise, and you compute the so-called grid score, roughly an order parameter for hexagonal grids, the distribution of grid scores here and here are indistinguishable from each other. That's these blue and gray histograms here. But if you look at the grid scores from this data, you know, there's a heavy tail of real grid cells, hexagonal grid cells. Yeah. Oh, sorry. Yeah. In the model or in the brain? Yeah. Oh, yeah. So basically what you do is you look at you quote unquote record from a neuron inside your recurrent network, and you compute a place normalized histogram of firing rate, right? So red means this particular neuron was firing a lot whenever the agent was here. Blue means it wasn't firing a lot. You know, blue here means it wasn't firing a lot whenever the agent was there. So red indicates where the neuron was firing, blue indicates where it wasn't. In this two-dimensional arena. So we literally have a two-dimensional arena. We simulate a mouse's motion. We make it undergo an Orstein-Uhlenbeck type random walk. We train the neural network to output a place code for velocity inputs. And then this is what the internal representations of this network look like. These fire, you know, so this is like one neuron, this is another neuron, this is a third neuron, and so forth. Yeah. Yeah. Yeah, those are the actual locations. So we feed the network the correct position of the agent through a place cell code. No, yeah, it's the actual position. Yeah. So that's just a method for getting a network in an unbiased way. Is the what? The J is trained. Yeah. Just back property time. Yeah. Yeah. Oh, sorry. There are two. Yeah. M is a 2 by M matrix, or N by 2 matrix. Yeah. All the nodes are connected to all the nodes. Yeah. 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 These are the targets. Yeah. They only have a single active, single bomb. Yeah. Yeah, the internal representations of individual neurons as functions over space, their firing rates as a function over space are not single bumps, they're multiple bumps. But the output is single. Every individual neuron in the output has a single bump. Yeah, yeah. So here, so here's the nonlinearity. This is a tan H nonlinearity. Yeah. Yeah, it'll look like this. Yeah, yeah. So the key issue is non-negativity. And by the way, we have a theory of all of this. We don't have to be mystified by this result. I'll explain all of this mathematically soon. Good thing I skipped the retina. It's so boring. Yeah, Paxton. Yeah, it's a stable activity pattern. Yeah, there's lots of fixed points in this network. I'll show you. No, it doesn't. You don't need to satisfy Dale's law. You just need non-negative firing rates. You could train it with Dale's law and it'll also work, but it's not necessary. Yeah. Yeah. Hopefully in a good way, not a bad way. Oh, did you have a question? Oh, okay. All right. Okay. Okay. Yeah. Play cells. But I don't think the play cells in the brain are playing the role of play cells here. But, yeah. We do get multiple scaled modules, but for an art, I think it's for an artifactual reason. So I'm not going to make a big deal out of them or even talk about them. Yeah, so you get heterogeneous. Actually, let me show you the entire data set, right? So these are the highest scoring grid cells in the population. So we had about 1,000 neurons in the hidden neurons in the network. These are the highest scoring grid cells. These are lower scoring grid cells. So you get grid cells and heterogeneous cells all together, just like in the brain. You know, that's actually a very hard thing for theorists to build. How do you build a circuit that has pristine hexagonal grid cells and heterogeneous cells? They're interacting with each other, but the interactions don't destroy the order or the randomness of each population. So order and randomness coexist in this network. I actually don't know how to do that from scratch. But these networks naturally do that. And we compared the structure of the heterogeneity in this network quantitatively to the heterogeneity in actual neural recordings in entorhinal cortex, and we got a really nice match. I don't have those slides, but it's in the paper. Okay, anyway, so these are so these aren't just, I didn't cherry pick some 6 examples here. This is kind of typical. And then, okay, now here's the theory, all in one slide, okay? So let me work through it. It will take some time on this slide. Okay, I think we can get it done in a few minutes, okay? So what determines the structure of internal representations in the network? Well, what a network develops in these internal representations depend on what you ask it to spit out in the output. Okay? And what you ask it to spit out, and in particular, how does it depend on what you ask it to spit out in the output? It depends on the correlational structure of the neural code of the output. By that, I mean, if the mouse is at a certain position, then there's a desired vector of activity across neurons when the mouse is at that position. When the mouse is at another physical position, there's another vector of activity of desired output at that other physical position. I can take the dot product of these 2 vectors and I'll get a position by position matrix, a correlation matrix. Okay? I can't show you a 2D position by 2D position correlation matrix, but what I can do is I can take the neural representation when the mouse is at the center of the arena and take its dot product with the neural representation, desired neural representation of the output when the mouse is at other points in the arena. And I can plot that dot product as a function of 2D space in the arena. This is like one row of the 2D by 2D correlation matrix. Okay? There's a second thing that we need. We need this correlation matrix to have a center surround structure. I'll explain why. Okay? So this is the core. So roughly you can think of this as the core translation invariant correlation structure of the desired output code, okay? The network has to build internal representations such that it can spit out this correlation structure in one synaptic step. Okay? So how does it build up those internal representations, i.e. functions over 2D space? Well, we can look at the eigenvectors of this matrix. It's a Toeplitz matrix. So its eigenvectors are roughly, because it's roughly translation invariant modulo the boundaries, its eigenvectors are just Fourier wave vectors over 2D space. So this is the Brillouin lattice, or the wave vectors in 2D space, right? And so the eigenvectors are Fourier modes in 2D space. I've arranged the eigenvectors associated with their their wave vectors. And so these are the low-lying eigenvectors of this spatial correlation matrix of the output. Similarly, for each pair of wave vectors, I have an eigenvector and associated eigenvalue. The eigenvalue is just the power spectrum of this image here. Okay? So if you have a center surround structure, then there's power in the eigenvalues along a ring or annulus of wave vector modes, right? Okay. So all I've talked about is the correlation structure of the output and its eigenmode structure. Okay, why did I belabor that point? It's because the internal representations of neural network, according to our theory, which I'm skipping a lot of steps, are constructed from the eigenmodes of the output, it turns out. In fact, the dominant eigenmodes of the output dominate the internal representations. Now, in this setting, you have a degeneracy of many possible eigenvalues or eigenmodes to choose from. Okay. Now, let's review how grid cells are constructed in the Fourier basis. Okay. Let's say I construct a function over 2D space that just has a horizontal wave vector and a vertical wave vector in superposition. Then I'm going to get a square grid cell. If, on the other hand, I choose a random linear combination of all of these eigenmodes, i.e. these things, then I'll get a heterogeneous grid cell. On the other hand, if I choose 3 wave vectors whose 3 modes, eigenmodes, whose wave vectors sum to 0, I'll get a hexagonal grid cell. Okay. If this doesn't have a center surround structure, it's just a localized Gaussian of large width, then by the uncertainty principle, the dominant eigenmodes will be near the origin, and the lattice points that intersect this will be along the principal axes, and you get square grid cells. And that explains the results in Cueva and Wei here, because they didn't have a center surround structure in that paper. On the other hand, in the DeepMind paper, if you don't have a non-negativity constraint, you have access to all of these modes, right? And so you can construct anything you want by taking linear combinations of these modes, and you get these heterogeneous grid cells. So that explains this result. Okay. On the other hand, if you have non-negativity, what happens is you can think of the non-negativity as promoting only positive activity patterns, right? If you're promoting positive activity patterns, that you're favoring an asymmetric term in your cost function, let's just Taylor expand it to be a cubic term. Now, let's say that you're trying to maximize the projection of internal representations onto these eigenmodes, but you also have a cubic term to force positivity. Then that breaks the degeneracy of these eigenmodes and picks out wave vectors that preferentially sum to 0 in Fourier space. And then you get hexagonal grid cells. Okay? This can all be spelled out, mathematically, but it's related to Swift-Hohenberg equations with a cubic term. And so that explains why we get these different properties. Okay, how do these networks work? Let me just, we have kind of 1 minute left. We can analyze the nonlinear dynamics of these neural networks and ask the first question we'd ask about any nonlinear dynamical system: what are its fixed points? Okay. It turns out its fixed points form a toroidal attractor manifold that are in one-to-one correspondence with points in physical space. And if the mouse is stuck at one location, neural activity will be stuck in firing rate space at one of these fixed points. And as the mouse goes round and as the mouse explores the space, the activity will go round and round the attractor, yielding this grid-like pattern here. How do these networks work? Well, what we can do is we can take these neural networks and disentangle them. Oops. We can disentangle them. And we can actually, we figured out an algorithm to place these neurons on a neural sheet. And we can look at activity patterns of this neural sheet. And we see when the mouse is at one instant of space, there's a hexagonal bump of activity along the neural sheet. And as the mouse moves, this hexagonal pattern moves along the neural sheet. This is exactly how theorists can design detractor networks in older work, right? However, in the older work, you could see the structure of the connectivity in individual neurons. You can't see it here. You have to do this collective analysis, and I won't go into the details of that. But basically, we can understand the connectome and the neural activity patterns and how they work. And so that kind of explains everything. Oops, yeah, okay, so I'll stop here. Thanks. Yeah, we asked a lot of questions. I've had, yeah, Fritz. Right. But I just have a question. Does anyone know where the plug is? Oh, okay, thanks. Oh, it probably won't. Okay. Don't worry about it. Okay, well, we won't need the slides. Yeah. Sorry, go ahead. Yeah. No, they don't. They're just a teacher signal in our model. No, the place cells don't feed back to the grid cells. It's unidirectional. So yeah, this W is unidirectional. So the grid cells are in the J. Yeah, exactly. Yeah. The integral of log x is going to be hard because I can't record from your brain when you're doing that. But I mean, there's a whole branch of neuroscience, motor neuroscience, that looks at relationships between motor cortical activity and muscles. We've actually done some of that work, and people are starting to understand that mapping. So, you know, if you put together, like, there's a whole bunch of studies that study the same behavior, like go, no-go tasks or 2 alternative force choice tasks. And across all of the labs across the world, they've recorded from multiple brain regions. And so, you know, you have to put that studies together. There's a lab called the International Brain Lab, where you have 10 different experimental labs recording from combinatorially many brain regions while the mouse is doing the same task. So you have to put all of that together. It's very difficult to record from the entire brain during one task. So there are some, you can do that in flies, for example. And so that might be a good place to try to understand that question. Yeah. By the way, we do have a paper that just appeared in Nature recently with Mark Schnitzer, where we could record from 8 different brain areas while the mouse is doing a choice task. And we see a very interesting thing where when the mouse is making its decision, that decision is broadcast to the entire sensory part of the brain before the mouse makes a motor movement. So it's as if the decision-making area is telling the sensory area, hey, heads up, I'm about to do this. And then it does it, right? So there's all sorts of interesting stuff going on in that respect. Yeah. 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 So the short answer, it sort of does, but for an artifactual reason, it is like if you pick out like the effective wavelengths of these low-lying lattice points, they obey a particular multiplicative ratio. But I don't think that's the reason the brain has modules. So it turns out you do sort of get loose modules, but I don't want to make a big deal out of it. I don't even want to talk about it, to be honest. Yeah. I think we have to do more work to explain the module structure. Yeah. Yeah. I mean, one of the things is like, imagine a downstream region that wants to read out the identity of the stimulus. If it had to record from 10,000 neurons, listen to 10,000 neurons upstream, then you need an awful lot of connectivity. So maybe the bottleneck is the limited connectivity in the brain. And you just need to get it so that any random set of 1,000 neurons has all the information, and packing more information into more neurons doesn't help because you don't have enough connectivity to transmit it. Maybe, but that's a just so story. Yeah. Yeah, I skipped that part, but these are the principal eigenvectors of that connectivity. It has a center surround structure when you average over all the neurons on the 2D sheet, when you average the displacement between the connectivity between one neuron here and one neuron here, fixing the displacement but averaging over all the neurons, you get this structure. And this is what you get in a very clean way for individual neurons when you hand design the network, but you don't get it in a clean way for individual neurons in this network. You only get it when you average over all the neurons. So it's an emergent property of the circuit when you train the network, but it's hand designed at the single neuron level when you forward engineer the network. But the principle is the same. Right? Oh, Mark, what's up? Yeah, 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 yeah. Um, yeah, I mean, you can imagine that. I mean, the short answer would be that entorhinal cortex is not in this dynamically critical state. It might be in an error correcting state, right? So, I mean, that would be like a just so story to explain the discrepancy. Yeah. There's no reason that every network has to be in that state. It's just an empirical observation that the visual cortex is in that state. I mean, I suspect so, because if the entorhinal cortex, if its function really is to path integrate, if it were incredibly sensitive to small inputs, it would be really bad at path integration. So it has to have an error corrective capacity in order to do its job. Right? Yeah, yeah. I mean, in the phase diagram, if you just want to assume sort of random connectivity with a ratio of excitation inhibition, then if you tune the ratio of excitation emission appropriately, you'll get away from this critical point. And the critical point is a very special point, right? You know, you have to tune something to get there. Yeah. Oh yeah. um, we haven't, but my colleagues Arseny Finkelstein in Israel, I forget who is the person who does the bat work in Israel. Ulanovsky, sorry, I so temporarily blanking his name. They looked at head direction of a bat in 3D space. So then your configuration space has the SO2 SO yeah SO3 symmetry, And so you're on the sphere, and so you have non-commutative action of SO3 on the sphere. But they actually didn't find, a nice prediction would be that you'd get spherical harmonics as your tuning curves. You don't actually. It factorizes space into like I forget which one's which, yaw or roll or whatever, and then yeah, there's some weird factorization going on there. So it's and I don't believe there's a good theory for why it's like that. That would be very interesting to think about. You have a question? Oh, okay, yeah. So the center surround structure of the desired place cell code plays a critical role, because I lost my computer, but what happens is you get high frequency modes that are those are the only ones available to you. So then if you take a triplet of high frequency modes that whose wave vector sum to zero, you're stuck with a hexagonal grid. And the wavelength is determined by the wavelength in the output, right? So because the place cells are narrow relative to the width of the environment, you must get multiple grids in the internal code. If your place cells were wide relative to the width of the environment, then you may not get multiple grids in the internal code. But that's like shrinking the ring to the lowest frequency modes. Oh, hey. Yeah, it's a great question. I don't know. I mean, it's the most generic solution, right? Like the pristine hand design connectivity is a very non-generic solution. It does probably have the advantage that it minimizes wiring length. So I think that's a good advantage of the pristine connectivity. And I know in the system that you're looking at, actually, do you know if it's local connectivity? Yeah. If you haven't met Yvette, she's a relatively new faculty who's working in beautiful stuff on the head direction system of the fly, which is like a one-dimensional version of the grid cell system in the mouse, and it's much more tractable. You can do amazing experiments. You should all talk to Yvette. And also Mark Plitt is you're a big boss now, Yvette, so Mark's doing all the work. All right. um, yeah. Yeah. Oh, sorry. Go ahead. Oh, Frederick, go ahead. Yeah. That's the hardest part. Yeah. So it has to do with like, so what you can describe the neural representations as trying to find principal eigenvectors of the spatial covariance matrix. So it's doing PCA on the output place cells, but with a non-negativity constraint. So the PCA optimization problem, like let's say G is the grid cell and C is the correlation matrix. The principal eigenvector problem is maximizing G transpose CG, right? But let's say you add a cubic term, G cubed in there, which if you're maximizing, that's forcing G to be positive, right? So when you analyze that optimization problem, G transpose CG plus G cubed, you find that the first order effect of the additional G cubed is to restrict yourself to, when you transform it to Fourier space, is to restrict yourself to combination of wave modes of large eigenvalues that also sum to zero as triangles. And the triangle comes from the cube and the G cubed, right? Yeah, so it shouldn't be obvious, but it's actually a very well-known thing in pattern formation theory. Huh? The distance between the which dots? Sorry, oh yeah. There's one wavelength. There's essentially one overall wavelength because you're taking 3 Fourier modes on an annulus of a given radius. So the wavelength of the entire system is determined by that radius. Yeah. Exactly. Yeah. 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 Yeah, well, in these trained networks, the pattern scales as well. In actual mice, what happens is there's the pattern eventually scales, but there's a transient. And there's much more effects going on, like borders can correct the grid cell code. And so we're not modeling any of that stuff. We're not modeling border cells. But what's really cool, there's a whole phenomenology in this field. Like what's really cool is if you make an irregular environment, like a triangular wedge or something, the grid cells will bend, right, towards the bend in a certain way. And we can explain that too in another paper where we hand design a network with grid cells and border cells correcting the grid cell code. And because the borders are irregular, the border correction will bend the grid cell firing fields. So we're not putting in border cells into these simulations, but we have them in other hand design models. So there's all sorts of stuff going on there. Yeah. 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 That's a good question. We actually haven't done the test of robustness to Gaussian noise, but I would be, actually, I don't know whether it would be more or less robust than hand design networks. We should do that test. Yeah, I don't want to say, I don't want to venture a guess. Yeah, but great question. We'll try it. Thanks.